So mitosis and cytokinesis is actually one part of the entire cell cycle. So ito yung cell cycle. The M phase or mitotic phase is actually 10% of the cell cycle. And this includes the prophase, metaphase, anaphase, and telophase that we have learned during our high school biology. And the M phase is usually the shortest part of the cell cycle, and it alternates with the longer stage called interphase. So ito yung interphase, and it accounts for 90% of the cell cycle. And it is divided into three phases. So first we have the G1 phase or the first gap. Then we have the S phase or the synthesis phase. And the second gap or the G2 phase. And a human cell actually completes its cycle for about 24 hours. And yung M phase natin, isang oras lang usually. And yung S phase, dito nangyayari yung pag-duplicate ng DNA. So from unduplicated chromosomes, di ba? Nagdo-duplicate sila. And yung S phase natin, nag-occupy siya for about half of the entire cycle. So kung yung entire cycle natin, 24 hours, usually 10 to 12 hours yung S phase o yung pag-duplicate ng DNA. Again, isang oras lang usually yung mitosis and cytokinesis. And yung buong cell cycle is 24 hours sa human cell, okay? So what happens during G1 phase or the first gap? So nag-i-increase yung size ng cell and then protein synthesis. The cell actually synthesizes different proteins na kailangan natin for S phase. And the organelles produced are in large quantities. So kailangan din mag-duplicate ng mga organelles, hindi lang ng DNA, okay? Next, we have the S phase. DNA replication happens during S phase, where each chromosome is duplicated. And one chromosome now has two sister chromatids. So ito yung unduplicated chromosome during G1 phase. And after S phase, duplicated na yung mga chromosomes natin. So kung yung isang unduplicated chromosome, isa lang yung chromatid, after DNA replication, meron na siyang pair of sister chromatids. So dalawa na yung chromatids niya. The primary event in S phase is the replication of DNA. Next, after the S phase, we have the G2 phase or the second gap. And it is similar with what is happening in G1 phase. So there is more cell growth. There is more protein synthesis happening. And dito rin nagpe-prepare yung cell for cell division. Kasi after ng interphase, after ng G2 phase, mag-undergo na siya ng cell division. So during G2 phase, chine-check din ng cell kung merong mga errors sa pag-duplicate ng DNA. Kasi hindi may iwasan yung mga errors kapag nire-replicate yung DNA. So dito, mini-make sure na okay yung DNA natin bago siya mag-undergo ng cell division. Okay? So ito yung itsura ng cell during G2 phase or before siya mag-undergo ng mitosis. So as we can see here, nandito yung mga centrosomes natin. Na-discuss natin in the previous video na yung centrosomes natin, they are composed of a pair of centrioles. And then meron tayong nucleolus, and this is actually the nucleus. And we all know that nucleus is enclosed in a nuclear envelope. And yung mga chromosomes natin, duplicated sila pero hindi pa sila nagko-condense. So, kailan sila magko-condense? Sa mitosis. Okay? So, after the interphase, ito yung pinakamahabang part ng cell cycle, mag-undergo na siya ng M phase. So, yung M phase natin is composed of the prophase, metaphase, anaphase, telophase, and the cytokinesis. So prophase, what happens during prophase? So chromatin fibers of the chromosomes condense. So before siya mag-undergo ng mitosis, ganito pa yung itsura ng chromosomes. Parang kalat-kalat pa sila, mahaba, long, thin chromatin fibers pa yung mga chromosomes natin. Pero kapag prophase, magsa-start na silang mag-condense and medyo visible na rin yung sister chromatids sa bawat chromosome. Kaya yung clue natin sa prophase is letter P, pair. So makikita natin yung pair ng sister chromatids during prophase and visible na sila under the microscope. And the nucleolus disappears. So sa G2 nandito pa yung nucleolus pero sa prophase wala na siya. And nagsa-start na rin mag-form yung early mitotic spindle sa ating centrosomes. So as we all know, the main function of centrioles is mitotic spindle formation. After prophase, meron tayong additional step na pro-metaphase. The nucleus is enclosed in a nuclear membrane. So para makawala itong mga chromosomes natin, the nuclear envelope needs to disintegrate para magkaroon ng access yung mga microtubules natin sa mga chromosomes. And then the chromosomes become more condensed. So tingnan natin yung difference. Mas naging short sila, mas naging condensed. And during prometaphase, microtubules attach to kinetochores. So ano yung kinetochores? Ito yung mga protein structures ng mga centromeres sa bawat chromosome. So yung mga microtubules natin from the centrosome begin to attach to the kinetochores of the centromeres of the chromosomes. Okay? So again, during prometaphase, nuclear envelope disappears, chromosomes become more condensed, and the microtubules from the centrioles attach to the kinetochores of the chromosomes. During metaphase, chromosomes align at the metaphase plate. The metaphase plate is an imaginary line that is equidistant from the two spindle poles. The centrosomes are now at the opposite poles of the cell. So nandito na sila sa magkabilang dulo. And the clue for metaphase is letter M. So we have the meeting of the chromosomes in the metaphase plate. So magmi-meet na sila sa center. Okay? So M, metaphase, meet at the center. After metaphase, we have anaphase. During anaphase, sister chromatids are now pulled apart, and each chromatid becomes an individual chromosome. Anaphase is actually the shortest stage of mitosis. And makikita natin na nagsiseparate na yung mga chromatids. So the sister chromatids are pulled apart by the spindle fibers. So each chromatid is now considered an individual chromosome. So dito, ilan yung chromosomes natin? We have 1, 2, 3, 4, 5, 6. So each chromosome here has 2 sister chromatids. Pero dito sa anaphase, magse-separate na yung mga sister chromatids. So bawat chromatid, isang chromosome na yan. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So from 6 chromosomes, naging 12 na yung chromosomes sa cell na to. So because the microtubules are attached to the centromere region at the opposite poles of the cell, the chromosomes are pulled apart. And makikita natin dito na medyo nag-elongate din yung ating cell to help separate yung mga chromatids further. Okay? So the clue for anaphase is letter A. So apart. So sister chromatids are pulled apart. Again, from 2 sister chromatids in each chromosome, naghiwalay na sila, and each chromatid is now considered an individual chromosome. After anaphase, we have the telophase. So telophase, during telophase, chromosomes decondense and the nucleolus reappears here, and the nuclear envelope reforms. And the mitotic spindle disassembles. So mawawala na itong mga mitotic spindle. So in telophase, the division of one nucleus into 2 genetically identical nuclei is now complete. So the clue for telophase is letter T. We have 2 genetically identical daughter cells. So from 1, naging 2 na sila. Okay? 2 daughter cells. So we started with 6 chromosomes. So each nucleus will also have 6 chromosomes. So 1, 2, 3, 4, 5, 6. So 6 dito and 6 din dito. Okay? After mitosis, cytokinesis happens, and this refers to the division of the cytoplasm. So the 2 daughter cells appear shortly after the end of mitosis. Cytokinesis is actually different in animal cells and plant cells. So in animal cells, cleavage furrow forms. Cleavage furrow is actually a shallow groove that is composed of actin microfilaments. And nag-i-interact yung mga actin na to with myosin molecules na nagkakos ng contraction sa isang dividing cell. So para siyang drawstring na nagpupull para mahati yung cell into 2 daughter cells. Okay? So again, in animal cells, cleavage furrow forms, which is composed of actin microfilaments interacting with myosin molecules. So for plant cells, cell plate forms. So as we all know, plant cells are composed of cell walls. During telophase, vesicles derived from the Golgi apparatus move along the microtubules to the middle of the cell. So these are actually vesicles, okay? So pumupunta sila sa center, forming a cell plate. And this cell plate enlarges until its surrounding membrane fuses with the plasma membrane. So itong cell plate na to, eventually magiging cell wall siya. So mahahati yung cell into two. Again, as a recap of the M phase, so prophase. So ito yung itsura ng cell under the microscope. So the chromosomes are condensing and the nucleolus is beginning to disappear. And kahit hindi pa siya visible dito, nagsistart na mag-form yung mitotic spindle natin during prophase. And then during prometaphase, mas visible yung chromosomes natin and mawawala na yung nuclear envelope. And sa metaphase natin, yung clue natin is meeting at the center. Kompleto na yung spindle fibers natin and magsistart na mag-attach or mag-align yung mga chromosomes natin sa spindle fibers and mag-align na sila sa center or sa metaphase plate. And sa anaphase, apart. So letter A. So chromatids of each chromosome have separated. So magsiseparate na sila dito. So the daughter cells are moving to the ends of the cell as their kinetochore microtubules shorten. So para silang hinihila ng mga microtubules. And in the telophase, daughter nuclei are forming. Cytokinesis has started. So since plant cell to, cell plate yung ma-form. Okay? So the cell plate which will divide the cytoplasm into grows toward the perimeter of the parent cell. Kaya yung cell division na nangyayari sa prokaryotes, for example, in bacteria. So prokaryotes, for example, archaea and bacteria can undergo a type of cell division in which the cell grows and then divides to form two new daughter cells. So tinatawag natin yun na binary fission or division in half. So binary fission, yung nangyayari na cell division sa prokaryotes, and sa eukaryotes, we have mitosis. Okay? So sa bacteria, yung pag-replicate ng DNA niya nangyayari sa isang origin. So chromosome replication begins at the origin. Soon after, one copy of the origin moves rapidly toward the other end of the cell by a mechanism involving an actin-like protein. So replication continues. One copy of the origin is now at the end of the cell. So meanwhile, the cell elongates. So again, yung DNA replication sa bacteria, nasa-start siya with an origin. So parang hinahati yung strand ng DNA molecule sa prokaryotes. Tapos, from one copy, nagiging dalawa. And after replication, the plasma membrane is pinched inward by a tubulin-like protein and a new cell wall is deposited. So proteins cause its plasma membrane to pinch inward, resulting in two daughter cells. Through binary fission, each cell inherits a complete genome that is identical to the parent cell. Cell division functions for asexual reproduction in prokaryotes and in some single-celled eukaryotes. And cell division also functions for growth and development and repair of damaged cells. So mitosis is really important for the proper functioning of living things. Okay? So that ends my discussion for cell cycle. Thank you very much for listening.